Thank you. Thanks for coming. Thanks for spending a few minutes with us to take a look at these exciting opportunities for bioprocessing. A little bit, I'm sure that you I'm aware, but Thermo Fisher is very strong in the environment, so making sure that our tagline is really making the world healthier, cleaner, and safer through our products, our services, and our technical capabilities, really to enable our customers to drive towards a healthier, cleaner, safer environment while they manufacture and support biotechnology and pharma. I have limited time today, so I'm going to clip through this pretty quickly. There's quite a few slides here. I'm going to talk about the commercialization from early phase discovery through large-scale scale-up. I won't go into any details there, but I want to highlight as you think about your development process, what are the precepts you want to think about? So often folks will not have in mind the big picture in the long range. So you start thinking about, I have a seed, I have a cell line, I want to do some expansion. One needs to keep in mind where you're going with the ultimate commercialization. So I'm going to talk about that a little bit and what those capabilities are. Just want to highlight here that at Thermo Fisher, we have a very strong capability of research and development. We do process development, early optimization. We have a pharma services group, Patheon, our legacy Patheon is a CDMO, as well as we have support capabilities for you at your sites to do early and late stage development and commercialization, as well as helping you build new facilities, should you be interested in that. So today, talking about solutions built for scale. So I was talking about early cell culture development all the way through bulk freeze and scale up. Really, I want to go through the gamut with you of when you're developing a process, you want to think about the outcome. So I'll give you a little trivial thing. When I worked at Genentech Roche prior to this for years, and we were developing bulk freeze applications. And normally at the R&D or development stage, folks will be developing something like this size, or let's say 1 or 2 liters, and they throw it into a Revco freezer. That's not scalable. And you have to start thinking about what you're doing at the early stage, especially when you have stability, validation, quality, and filings. And really, we believe that we have very unique capability to scale all the way from small scale, early stage development, all the way through commercialization, both in the cell culture, upstream, and downstream areas. So let's talk about map processing with regards to this precepts I just spoke about. Here is a flow of our monoclonal antibody production, and shown on here is obviously not all-inclusive, but some examples of the unit operations that we sell, develop, support, both, again, to develop your processes in-house, which I'll talk a little bit about that at the end here for our new Bioprocess Design Center, which is purposely built for that, our CDMO, and or work with you with these technologies in your actual facilities, either in development or clinical or commercialization. So as you look here at the flow, don't think it works here. If you look at the flow here, you see that we have our single-use bioreactors and the capability. We don't just have single-use bioreactors, we have the most capable single-use bioreactors in the world. That's a fact. And the reason they're most capable is their turn-down ratio. So we have the ability to go from 500 mils in a stirred cell single-use bioreactor with the same sparging material, the same KLAs, the same constants, the same automation that you would use all the way at the 5,000 liter scale for commercialization. You can seed these reactors at 10 or 21 turn down, which allows you to skip steps. And I'm going to talk about that efficiency in just a second. Likewise, in downstream, we launched a couple of areas in protein purification, which are very novel. And what I'm going to do for the rest of this talk is highlight why those unit operations can drive huge economic benefits for you as you start thinking about development and going into commercialization. So you don't have to raise your hands, but I thought about this morning when I was driving down. I live up in Carlsbad through the traffic. How many people are going to aim, whether in early stage or late stage, for like maybe $300 per gram. $300 per gram sounds pretty good for mAbs, right? No, it's not. That's early stage, that's expensive. The industry in general is roughly spending about, if you write a contract with CDMO or you're trying to develop in-house, somewhere around $180 to $150 per gram would be average for a 3 to 5 gram CHO line expressing CHO line. What I'm showing here is a way for you, a way for industry, a way for mAb manufacturers to get to $50 per gram. So $50 per gram is a moonshot, right? So it's really something that the industry, so I've heard Microsoft, many of the manufacturers here, CDMOs, our competition, everyone's driving to that magic number. And so what I'm showing you here is how do you increase your titers, reduce your cost per gram, and then overall increase your profit. So I'm going to walk through some upstream and downstream examples how you do that, but keep in mind this needs to transfer from early stage development into commercial, and we have the tools to do that. If you need a copy of this, we can get into the details. I don't have enough time, so it's roughly an hour talk if you want to go into each of your unit operations. So we have publications on this as well. Some of the areas that drive intensification and monoclonal antibody processing are shown here, and they're highlighted, and I'll go through some of the case studies very quickly with you. High-density cell banking and N-1 perfusion. So what that means basically is that you're able to go directly into the single-use bioreactor and skip roughly 6 to 10 days of GMP manufacturing time. So normally you would do an expansion with a seed, then you go into shakers or spinners, then you go into maybe a rocker, and then you go into your stirred cell. We're able to work these bioreactors with our enriched media, our high-producing cell lines, and go directly into a 50-liter bioreactor, which then lets you expand into a 500 and go directly into 5K. And I'll talk about the return on that of investment in just a minute. As well, we have intensified fed batch protocols, which allow us to drive our titers in excess of 12 grams per liter now. And when you start doing the mass balance, it's a pretty large amount of kg of protein coming out of this large unit. And then lastly, we launched roughly a year and a half ago our single-use centrifuge, and this can negate roughly 70% of your depth filtration. So if you look at the 3 steps and you add up the savings in time, the manufacturing building space, the suites that you either are not going to build or you don't have to continue to validate and run the HVAC through, and if you're brownfield or greenfield designing that you can negate for the high-density cell banking, the intensified bioreactors, and then negating your depth filtration or dramatically reducing it, that's where a lot of the economic return's coming from. Other ancillary products that go along with the mAb manufacturing that we support are shown here, and you can see that, and I won't go into any detail, but we have our single-use division and our BCD team, which is part of the GIBCO line, have quite a few products that support the intensified processes that I'm talking about. I want to highlight that we did procure the Solventum team from 3M, so that's our FSD, the filtration division now that we added, and a lot of people still use depth filtrations. These are, and we have some publications, extremely high throughput, very well clarifying depth filters. We're extremely glad to add it to our repertoire for filtration. So let's talk about some of the returns now and use some case studies here for true examples. So I talked about high-density cell banking, and what that means is that you're moving the expansion of your seed expansion to a high-density cell bank. And so essentially what you're able to do is take the bag on the left, which has your 6 to 10 days of rolled-up expansion time, frozen, and directly inoculate it into the bioreactor. So we've shown this a number of times both at 50-liter and 500-liter direct inoculation, and that saves a number of weeks of GMP processing. In the other area that it saves is in your processing area. So if you look at the GMP area for a standard process, and then you look at the percent reduction by not performing those GMP operations and going directly from a high perfusion, high density cell banking, then you're saving roughly 25% on your facility cost, which, by the way, when you're doing your cost per gram, your facility costs are one of your main sources of cost per gram. So through BioSol, which we use to model all of this information, which is used by most of everyone in the industry, your most expensive cost per manufacturing is your production bioreactors. So everyone will show that data if you look into cost per gram and ROIs, your protein A, and then your facility costs. So what we did was look at those areas and look at where do we need to innovate and develop our unit operations to hit those big rocks to reduce cost per gram. So again, back to high density cell banking, reduce the number of operators, reduction in half the time from 18 days to 9 days for the actual going to the production bioreactor. So you're looking at roughly a 50% cost per goods reduction there. What I'm showing here, and I won't go into detail, if anyone's interested, come talk to me, I'll be here all day today, is we did a number of runs, both in our single-use bioreactors at the small scale all the way up to the 5,000-liter bioreactors, and we're just trying to show here that the lines and the expansions track very nicely. They're linear and they scale up very well. Here what we're showing is that you're capable of taking your 50-liter bioreactor and running the high-density cell banking directly and scale it up to a 5,000-liter bioreactor. So we're quite excited about this. We have a number of customers that are interested in deploying this technology now in the CDMO environment based on the cost per gram target that they're driving for. I mentioned early depth filtration. So in standard mAb manufacturing, you will always need to use depth filtration. You can use centrifugation, but you're still going to have to clarify your final harvest material. Still has enough particulates, host cell proteins, and lysed DNA that you're going to mess up your most expensive downstream bill of material, which is protein A. But you can reduce. Here we're showing 30%. That's being very conservative. We've shown many runs where we reduce up to 70% of the depth filters by using the single-use centrifuge and then still deploying the depth filtration after the single-use centrifuge. And shown here is a percent improvement over filtration, the water usage, and the labor hours based on the reduction in the depth filters needed. Ancillary processes along the downstream are shown here. So we launched our Dynachrome. I won't go into depth on Dynachrome. It's its own talk. Suffice to say, this skid is extremely capable, and it's able to do 50-to-1 inline dilutions. So what that means is that when you're doing your buffer prep for chromatography, you can do it at least 10-fold less. So it means that you can make your stock solutions a 5x or 10x, hook it up to our proprietary inlets in the Dynachrome, and then do inline dilution in your clean room, whether it's a single-use clean room or stainless steel clean room, and dramatically reduce your labor and your time in buffer prep. So let's talk about the next big rock in cost reduction. We launched our mAb Capture C. It's an extremely capable, very stable, highly competitive protein A on the market. So if you remember in the beginning talk, I said you've got your production bioreactors. This is your largest cost per gram contributor to the mAb process in upstream. Protein A is your largest cost per gram in downstream. Our protein A, I won't get into exact discussions here with cost with you guys, but it's extremely competitive, roughly 40% less than the leading mAb capture. It's base stable, has over 200 cycles capability, and because of that, you can see that the resin batch per cost is roughly 30% less per run over run for a mAb process. And then if you gang together the inline dilution, then you're getting roughly a 50% reduction in just your mAb processing step by using inline dilution and our mAb Capture C. And then one last case study, and then I'm going to roll it up for you, is our HIG. So we have a hydrophobic interaction chromatography column. It's been used for years. It's not new. This resin is new, but HIC is not new. But what's new here is our ability to use it in a negative flow-through manner and gang it along with our protein A. And what we've shown is we've had improved yields over the standard MAB process, faster throughput, and a reduction of roughly 30% in the actual manufacturing cost based on the flow-through, the time in the suite, and the resins that you have to pack for the column. So it's a big improvement in the downstream step. When you roll it all up, and this is the slide I started with, you roll it all up, you start looking at upstream. We're looking at somewhere around a 2.5 to 3-fold reduction in increase in your KIGs, and you're having roughly a 50% reduction in your cost per gram. So we took a standard MAB process from 247, so we did this, we actually scaled this to 5K and measured the return. We're taking the process that we did with our CDMO from around $250 down to $115 per gram. That's substantial. So if you're already starting at somewhere around the 150s and 180s, you can imagine you're going to get down to your 70s and 75s pretty quick. Of course, the industry's driving for the 50s. And you're not going to do that just at upstream, so you're going to have multiple steps to do that. We're able to increase our cell titers dramatically with our sparging, our KLAs, and our capabilities in our bioreactors. And then you could look at roughly an 800% increase just in upstream from those unit operational changes. Downstream, we talked about the reduction of the footprint of the building, the CapEx that you have to spend for buffer prep, and the personnel for doing inline dilution and creation of the buffers, as well as the reduced costs for resins and throughput. And you can see here there's a dramatic savings here. Lastly, I'm going to spend the last 3 minutes on this. So how do you do this? How do you implement this? You can do it yourself. You have a strong MSAT, you have a strong development team. Absolutely, you can do this. And you should. However, we just launched our Bioprocess Design Center in Plainville. And so the sole purpose of this center is to do exactly what I'm showing you. So we can, if you want to touch any of the products, you want to experience the products, you want to kick the tires, you want to actually run runs in the equipment. If you want us to help design a process from you, take it from pre-phase to phase, or they take it from phase 1 to commercial, that's what the center is designed for. And we're there to collaborate. We're there to work with you under NDA if you need us to do that. If not, does not need to be NDA. And then train your teams. If you just want to come in and do a column packing, you want us to come in and show you how to do the inline dilutions on the chrome skid, that's what these centers for. We have 5 centers. The newest one we just launched, which I'm talking about, is in Plainville, Massachusetts. It's our largest flagship in the US. We also have China, Korea, Singapore, and India. All of them have extremely like capabilities, with the exception of the United States, which has very large capabilities going up to 5K. So I'll highlight that quickly. We had our ribbon cutting for this a couple of weeks ago. It was exciting. A lot of us here were there. This center reports to me. We have some very strong PDMSAT type folks running the lab. It's a very nice space where you can come bring your team, bring your executives, talk about some of the processes I was just talking about. How can you get to the goal that you're looking for either scale up or financial or building your facilities? So we can do some modeling for you for those facilities, throughputs, clean rooms. What you're looking at here is our conference center, which then has windows which you can look into the actual processing area, and we have executive conference room that also has clear windows directly into the labs. Here's a little bit about the lab. We built this lab purposely like you would a GMP facility. And it's not GMP, it's a lab. You're able to come in there, go in with your jeans and just a lab coat and glasses. Everything's closed, gamma radiated. It's a true running lab, so we can actually not just demonstrate, but actually run your cells, your clones, help develop, optimize your process, scale it up from bench, all the way up to 5K, shown here on the right of the screen there. So we're super excited to have this capability and help optimize for you your processes and help develop and work on those. Again, a little bit of the processes that we operate there: process development, process optimization, both upstream and downstream. We can do media selection, feed strategies, filter screening, and downstream purification development. I think that's it. Thank you.